afternoon, everyone. My name is Vinny, and today in this big meeting, I will present about the recent advances in bioethanol production via fed patch strategy. Here is the outline. First is introduction, and then continue with the enhancing strategies includes the different feeding pattern, monitoring evolved gas, and the different feeding time and enzyme loading. So then continue with the summary, and lastly it's the references. So now the world energy source were predominantly by the fossil resources, which accounted by around 90%, and less than 10% is the renewable resources. And these renewable resources also predominantly by the food and feed crop based biofuels and biogas, accounted by 4%. Biofuel includes the bioethanol, which is renewable, biodegradable, low toxicity, and environmental friendly. And this is the trend of global bioethanol production, in which predicted will be increased over years. And because of this demand is increased, the market value will also increase. There are four generations of biofuel production, which is the first generation is from the edible feedstock, in which this can take over the food resources. So there is another called the second generation is the non-edible feedstock from mainly from the lignocellulosic biomass. And the third generation is utilizing the algae and other microbes that can be utilized as the biomass to produce the bioethanol. And the fourth generation is to do the genetic engineering and nanotechnology to the microbes so they can produce more liter of bioethanol. Okay. So there are three fermentation strategies that we know, right? So the first one is the batch fermentation, which is considered as closed system where the nutrients introduced at the need initial process, and then the final product will be harvested in the end of the fermentation. And the second one is the continuous or chemostat strategy, which is considered as open system because the nutrient inflow equals to the product and residual nutrient outflow. And the third one is the fed batch system, which considered as open system also, but they will add nutrients without removing the products. Okay, so why fed batch? Because the fed batch is known as like the control to control the feeding rate to prevent osmotic inhibition, in which osmotic inhibition can interfere the growth and ethanol productivity. So the objective of today's presentation is to compare innovative fed batch strategies to enhance bioethanol production. So let's move on to the first paper, which titled Continuous Bioethanol Production from Glucose-Rich Hydrolysate Hereinafter GRH derived from wet straw using the unique fed batch cultivation method in a bioreactor. In this study, they used the Pseudomonas mobilis, which is known as high glucose uptake and high ethanol tolerant. And then they also pre-treat the wet straw using acid and recycle it to produce more of the glucose. And then fed batch strategy, they use a different feeding amount. So in this figure, there are three strategies that they use. The first one is using 20% retained medium and 80% new, which shows that the cycle 1 to 10 showed stable glucose consumption rate or GCR, and the bioethanol bioethanol yield. The second one is the strategy 2, which using 50% retained medium and then add with the 50% new medium. So and shows the GCR and BEP were higher than in the previous strategy. And the third one is the strategy 3 using the 70% retained medium and add with the 30% medium. So in this case, because the retained medium is more higher than the other two strategy, so in cycle 5 onward is decline, shows decline trend, and then the retained growth because too much and it can lead to toxicity of the cells. The second figure shows the fermentation profile for each strategy, in which the figure 2AB using the strategy 1 and the 2CD using strategy 2, and this both strategy exhibit similar BEP or bioethanol productivity, but with this case they generate faster cycle. And the third strategy, in later cycles, glucose disappear more slowly and the ethanol rise more slowly, as shown as in this picture, because it is really significant. So it's clearly shown as depicted. So the highest and stable strategy is the second strategy, which is 50% retain and 50% new medium. So therefore it applied to the glucose from GRH. So this is the fermentation profiles of the fed batch fermentation with GRH glucose, which is the fed around 30 gram per hour. And the AB figure shows that the across cycles 1 to 10, the GCR stable around 11 to 12, and the bioethanol productivity reaches 5.7 gram per hour. And then the C figure shows that the acetic levulinic acid concentration slowly rise from cycle 1 to 10, but remain below the levels that gave inhibition in the fermentation. And then the D figure shows the stable kinetics until 10 cycles despite on the increasing inhibitors. As you can see here, it is stable, but the inhibitors are still like rising over time. So let's move on to the paper 2, which titled an adaptive continuous substrate feeding strategy based on evolved gas to improve fed batch ethanol fermentation. In this study, they used the Saccharomyces cerevisiae and glucose as the substrate. The strategy is using the adaptive based on the evolved gas that produced during the fermentation. So there are three conditions that they use here. So first one is the control condition, which is the fixed continuous feeding nitrogen. So from here to here, AD, and then the glucose is pumped at constant rate using the high nitrogen to carbon ratio medium. And the second condition is adapted continuous feeding with high nitrogen medium from here to here. And then the third one is the adapted continuous feeding with low nitrogen content of medium, so from here to here. And then adapted means that they add the glucose just when they monitor the gas signal. Okay, so let's see the cell dry weight, which is the as shown here. Adapted feeding has higher biomass than the control, which shows higher concentration here, and then the adapted feeding has higher glucose efficiency, in which shows here, stay near zero and accumulate later here, and has lower peak. So all treatments showed ethanol concentration up to 90 gram per L, and glucose accumulation starts around 100, in which where the ethanol reach 60 gram per L. And then evolved gas flow rate were higher in adapted treatments, which means higher metabolic activity. So continue with the figure 5, which shows the total glucose consumed per liter. That area under the curve are 115 gram per L, and for adapted, and 105 for fixed feeding. So which means that the adaptive is not only changing the timing but also changing the conversion behavior. And the last one is this is the overall fermentation performance that's can obtain the effects of the adaptive strategy, which is the lower residual glucose, which means efficiency and increase the biomass, and then ethanol productivity increase, which means also the faster process of fermentation. So let's move on to the third paper titled Fed Batch Strategy Achieve the Production of High Concentration Fermentable Sugar Solution and Cellulosic Ethanol from Pretreated Corn Stover and corn cob. In this case, they use the Saccharomyces cerevisiae also, but they add enzyme to enhance the fermentable sugar from those cellulosic substrate, and then they use the simultaneous saccharification and co-fermentation. And this is the figure when they add 50% solids and 3 hours precerification at 48 degrees Celsius, and they add around 10 FBU of cellulase and drop the temperature to 35 degrees, then inoculate the yeast. And then the fed batch, they add solids at 3 hours and 6 hours to reach 25% and then 35% solids. And here, as you can see, ethanol concentrations rises quickly from 24 to 72 hours, then slowly slow. And then glucose was almost exhausted and at 72, and then the xylose and cellobiose declined gradually but remained until reach 144 hours. And the final ethanol titer is the 46.5 gram per L at 144 hour. Next one, they also use the same pre-saccharification to this CCR or the corn cob one, but they use less cellulase and drop and then also drop the temperature to inoculate the yeast and the fed batch at solids at 4 hours and 9 hours of to reach 23% and 29% solids respectively. So in this case, as you can see, the ethanol increased gradually from 48 to 144 hours, and the final ethanol is higher than the previous one, which is 72 at 144 hour, and then the higher ethanol titer is due to the higher glucan and lower lignin of the corn cob, and which this corn cob is better substrate in this setup. So the summary is feed batch enhance production by not accumulate big amount of medium in initial stage, so it gives less stress to cells. And fed batch strategy, including feeding concentration time and monitoring, may improve productivity and prevent accumulation of toxic and inhibitors. And then integrated fermentation approach provide a sustainable and scalable solution for large-scale bioethanol production, especially with lignocellulosic biomass. So here is the 3 references, and thank you for listening.